Hello everyone, my name is Arunabha Mitra. I'm a postdoc based at University of California, Riverside, working with Dr. Georgios Virlakis. And in this talk, I'll be presenting findings from our field trial investigating the biology of citrus yellow vein associated virus. CYVAV has had a long history at UC Riverside, dating back to 1957 when Professor L.G. Weathers encountered it in limecod trees. Until recently, however, it had only been described mostly as a mild pathogen that's easily graft transmissible to a wide variety of citrus cultivars, but not known to cause any major economic damage. CYVAV's genomic characterization was first published by our team in 2021, describing an approximately 2.7 kilobase per single-stranded RNA genome encoding two main open reading frames. It was subsequently classified as a class 2 umbra-like associated RNA or a ULA-RNA, which is relatively rare among citrus-associated pathogens. This small genome makes it intriguing as it lacks many of the conventional genes expected in other plant viruses. The photos on the right show foliar symptoms of CYVAV, both from the original Weathers paper and recently from our field trial. Given its mild nature, there still remain important questions about CYVAV, whether it poses a quarantine threat and how it might spread, questions best answered in a full-scale field setting. This research project consisted of two main objectives. First, we wanted to see if in a field setting CYVAV truly has any negative impacts on tree vigor, fruit yield or quality when introduced into a variety of commercially important citrus cultivars. Second, we wish to explore pollen as a potential mode of natural CYVAV transmissibility in the field setting. The overall goal was to gather tangible data-driven findings about CYVAV biology under field conditions. Towards accomplishing Goal 1, in October 2020, we planted a replicated field trial at UC Riverside, including 12 different commercially popular scion rootstock combinations, as listed in the table on the right. A month later, in November, we graft inoculated 6 trees of each variety with the wild-type CYVAV isolate YV920, being maintained in Mexican lime at the Citrus Clonal Protection Program. This was followed by regular monitoring of the field inoculated trees and an annual RT-qPCR screen for CYVAV infection. Preliminary observations with graft survival and infection rates led us to reinforce the initial inoculation with additional grafts, ensuring that the virus had ample opportunity to establish. In addition to quantifying the field CYVAV infection rate, we also quantified any potential CYVAV-driven tree impacts via measuring tree heights and fruit harvest data. We were able to collect 3 seasons worth of this data for these metrics. From 2021 to 2023, we tracked which inoculated trees became CYVAV infected via annual RT-qPCR screening. This table summarizes the symptom and infection rate results documented during this time. Many varieties remained near 0% infection by the third year, with Limonera and mandarins being the primary varieties that became infected with CYVAV. There was a gradual increase in infection rate documented over the 3 years of testing of these two varieties. Infected trees frequently had lower symptom rates, often half or zero, compared to the infection percentage, emphasizing that yellow vein symptoms are not consistent despite infection presence. We also documented instances of initial infection and graft survival, followed by consistent non-detection of CYVAV by RT-qPCR, highlighting very non-uniform virus titers across the infected trees. The grafts themselves generally survived well, with often 100% survival, but actual virus establishment varied, suggesting certain varieties being more recalcitrant to CYVAV colonization. Overall, after 3 years, CYVAV field infection rate was 28% among the graft inoculated trees. CYVAV was systemically detectable in infected trees. However, there were significant titer discrepancies, with many citrus cultivars seemingly resistant to CYVAV colonization under field conditions. Here, we compared the average tree heights of all 12 citrus varieties included in the field trial, measured from 2021 to 2023. The red bars represent inoculated tree heights, and the green bars represent untreated tree heights over the 3 time points. The big question was, does CYVAV impede infected tree growth? The short answer was no. We did not see statistically significant differences in height between infected and untreated trees. All varieties generally followed similar positive growth trajectories over the 3 years of data collection, with Limonera and Tango mandarins showing the highest growth rate irrespective of CYVAV infection. This data reinforced the indication that CYVAV, if present, does not reduce infected tree vigor under our field trial conditions. In this graph, we have a summary of the fruit harvested from the 5 CYVAV infected citrus varieties in this field trial, mainly lemons and mandarins, over 3 years from 2023 to 2025. Over the 3 seasons, there was massive increase in fruit yield for the Limonera trees, with no significant differences in yield between infected and untreated trees. In case of the mandarins, although there was a similar positive trajectory of yield increase, it was far lower than the lemons, which was expected. Even in the trees that were consistently CYVAV positive among these cultivars, yield metrics kept pace with the untreated controls. Again, there were no detrimental impacts of CYVAV infection that were apparent in these yield metrics. These photos on the right for lemons and mandarins provide a robust perspective on how both infected and healthy trees looked and bore fruit during the time of harvest. With each harvest, we also inspected basic fruit quality parameters. These included fruit color, shape, texture, firmness, sugar content or Brix, juice volume, juice weight, and titratable acidity. Summarized in these two graphs on the left are the juice volumes and Brix or sugar content from samples of 10 fruits per treatment, again from the 5 CYVAV infected citrus varieties from this field trial. The photos on the right represent the fruit samples that were tested, and visually you can see how the fruits from CYVAV infected trees, in case of both lemons, tango mandarins, and the Shiranui mandarins, appear almost exactly like the ones from their untreated counterparts. In case of both juice volumes and Brix, there were no significant differences between fruit from infected trees and their untreated counterparts. For mandarins, a Brix of 10 is commonly cited as an industry standard, which our trees, irrespective of CYVAV infection presence, were easily able to reach. This again aligned with our observations so far of no apparent detrimental effects of CYVAV infection, aside from mild leaf symptoms, at least in the infected citrus varieties here. Now on to our second research goal methodology. During our PCR-based screening, we saw that CYVAV was detectable by RTQ-PCR in pollen collected from infected field trees. Therefore, our second goal became to investigate pollen transmissibility in the field. So over 2 flowering seasons, we conducted artificial field pollinations in Limonera and Shiranui mandarin based on their pollen availability. For this assay, pollen was collected from both CYVAV infected and untreated field trees, and pollinations were conducted across 3 treatments. The test treatment was with infected pollen onto untreated flower stigmas, named YV on H. The positive control was untreated pollen onto infected stigma, named H on YV, and the negative control was untreated pollen onto untreated stigmas, named H on H. All treatments were performed within variety. This pictorial below gives a pipeline for how the artificial pollination was conducted for each flower. We start with a closed flower on a given tree, remove all petals and anthers, and then gently brush pollen onto the exposed stigma with a soft paintbrush, and finally cover the pollinated flower with a mesh bag and let it develop into a fruit. Overall, in 2022, 229 pollinations were performed, out of which 26 survived into fruit. And in 2023, 339 pollinations were performed, out of which 65 survived into fruit. Each year that the pollen transmission bioassay was performed, once the fruit set successfully from each set of pollinations, we harvested them in the following winter. Each fruit was then dissected with sterile razors into its pedicel, central column, rind, pulp, and seeds, if available, followed by individual tissue-specific nucleic acid extractions and RT-qPCR testing of each of these tissue types for CYVAV. This fruit dissection and RT-qPCR testing protocol was followed in both seasons of performing the CYVAV pollen transmission bioassay. Now on to the results of the CYVAV pollen transmission bioassay. These two tables summarize the total fruits for each treatment across the 2 varieties we tested based on pollen availability that successfully set from the field pollination experiments. As described earlier, all of these fruits were dissected and tested for CYVAV by RT-qPCR in a tissue-specific manner. We tested 26 fruits in the first year and 65 fruits in the second year. All fruit from the test treatment YV on H tested CYVAV negative, both years of conducting this bioassay. The same was also true for the negative control fruits. The only instance of CYVAV detection was in the fruit set on a CYVAV infected mother plant, but pollinated with healthy pollen, that is the H1YV treatment, which simply confirms that the mother plant was already carrying the virus. Overall, there was no evidence of CYVAV pollen-mediated transmission seen in this bioassay across both years of conducting it. Till present, the untreated trees remain CYVAV negative, despite the presence of local aphid species and natural pollination occurring every season. In conclusion, our findings indicate that certain citrus cultivars, particularly Limonera lemon and the Tango and Shiranui mandarins, were most field compatible for CYVAV infection, following multiple graft inoculations and extended periods of incubation under field conditions. Despite successful virus colonization in these hosts, we observed no detrimental effects of CYVAV infection beyond mild yellow vein symptoms on the leaves. Notably, the field harvest yield kept increasing from 2023 to 2025, with no significant differences in yield size or fruit quality between virus-infected and non-infected trees among the CYVAV-infected treatments, as well as the remaining scion rootstock treatments. Finally, although CYVAV was detectable in pollen from infected trees, our results from both years of performing the pollen transmission bioassay did not indicate any CYVAV pollen transmission. Untreated field trees remained CYVAV negative. Altogether, these results suggest that CYVAV, under our trial conditions, poses minimal risk to citrus health and production. I would like to thank the California Citrus Research Board for supporting this field trial. Please feel free to email me any questions at the email address shown here, or you could also direct them to my colleagues in the audience. I also thank the IUCV organizers for this great presentation opportunity. Finally, I want to give a massive thanks to Georgios and our CCPP team, along with the teams at the Lincove Research and Extension Center and the USDA for helping with the field data collection. It's a lot of effort and I greatly appreciate everyone who helped. With that, I conclude my talk. Thank you for listening.